Hello guys, welcome to my channel. So as you know, we are studying about the enzyme and we are continuing with the enzyme as well. So like this is the last time we studied, right? The enzyme, the function of the catalyst, right? And the ground state, transition state, and the energy, the activation energy. So now we'll see like the enzyme and substrate when they bind with each other. It's not that the substrate has a complementary shape like a lock and key mechanism, like it would bind and like it will start functioning or it will decrease the required energy, right? So to understand that, we'll go through some of the example. Right, so for example, we'll take a metal stick example. So for example, this is a metal stick. So this is a substrate. Alright, so this is a substrate. Now we have to break this metal stick to get the product. So like we have to bend this, right? So this, the bending of this metal stick is the transition state, right? And eventually the product, that is two broken part of the metal stick, right? So, right, the transition state. So this is the ground state, transition state, and then the final product, that is the final product, that is the ground state again. And the same diagram, for example, like this is the same diagram for this one, the first one, right? Now we'll see. So before the stick is broken, it must first be bent, that is the transition state, right? So now when we apply an enzyme to this, so we'll see the first example as a complementary shape. So complementary shape means exact, the substrate and the enzyme is the same as a same shape. So this is, for example, this is our substrate, this shape. So enzyme will be of the same shape. For example, this is, so this dotted, that's an enzyme. For example, this is an enzyme. All right. So this part is an enzyme and this is a substrate as before, right? All right, so this is a enzyme, right? So like we'll name as a Stick-Stickase. So that's a famous, like this model is really famous to explain the complementary shape. So this is a Stick-Stickase enzyme with the magnetic field, right, around the metal rod. Okay, so as this is a complementary in the structure to the stick, this stick as enzyme. So what happened? It impedes the bending. That is, it goes. So bending is impeded by the magnetic field or the attraction between the stick and the stick case, right? So it cannot go to the transition state and like there will be some product, but like a very less or few products. There will be very less product. Right, so and for this, if you see this kind of diagram, so like it will be like this is the S, and then the enzyme and the substrate complex will be down there, and then it goes to the transition state and then the product state, that is here. So you can see like this is substrate, so this from this part to this part, right? So this will be the delta G of the catalyzed reaction, and see how much energy is required to activate this to go into the product, right? So that's why complementary shape of enzyme and substrate is not so favorable, right? And this last example, that enzyme is complementary to the transition state, not to the shape of the substrate. For example, this is our same metal rod, right? And our Stick-Stickase enzyme is something like this. Like this, right? So this is something like this. So because of that, So when this metal goes into transition, that it bends, so the enzyme allows, right, and this will be the final product. This is the enzyme and we have the two final product, right? And for this, like it will be like this, the uh, enzyme substrate complex will be like this, and like it will go something like this, and then you will get the final product. So how much the activation energy is decreased, how much activation energy is decreased in this enzyme substrate product, and because of the enzyme catalysis. And this is how it should be, like the enzyme should be like this. It's if it's like complementary exactly to the shape of the substrate, then it won't allow a favorable reaction, and the product, you will get a few products with requiring more energy, right? So this was just an example for the enzyme complementary to the transition state. And this is like what happens in this. So the enzyme with the pocket complementary to the reaction transition state helps destabilize the stick, and that contributes to the catalysis of the reaction. What it does, the binding energy of this magnetic interaction compensate for the increase in free energy required to bend the, so this will pull it and it will help to bend the stick, and this is how it helps to increase the free energy. All right, so the real enzyme work on an analogous principles. So some weak interactions are formed in the enzyme substrate complex, but the full complement of such interaction between the substrate and the enzyme is formed only when the substrate reaches the transition state. Right? The full complement of the weak interaction, when it happens when the substrate reaches the transition state, that is this state or this state in the graph. So the free energy, that is the binding energy that will be released by the formation of this interaction, partially offset the energy required to reach the top of the energy hill. So the summation of the unfavorable, that is a positive activation energy, and the favorable negative binding energy results in a lower net activation energy. Right, so what does that mean? So, alright, so I will show you the graph here again. So, so this is a substrate, for example, and like it goes like this, and this, and this will be the final product. And if it's without the or uncatalyzed reaction, okay, so this is the substrate here, this you have enzyme substrate, and this is enzyme product, this is product, this will be the our transition, the top of the hill, right? So how the energy is, or the role of the binding energy in catalysis, so to lower, so this is the activation energy that you can see, right? If the reaction is uncatalyzed, right, uncatalyzed, so we can put uncat for the uncatalytic reaction, and for our G. Right, so if the reaction is uncatalyzed, right? So for example, this is the uncatalyzed reaction substrate to the final product, right? So it requires this much amount of energy, right? So when the catalysis occur, what it wants, right? The system must acquire amount of energy equivalent to the amount of this free energy, and it should be lowered. So then only the reaction will be faster. So much of this energy comes from the binding energy, right? So what is the binding energy? That is this energy. This you can see. So now as we have the substrate in the enzyme, so that substrate contributed the substrate contributed by the form weak non-covalent interaction between the substrate and the enzyme, and it creates a small hill. And so this here the substrate and the enzyme binds, right? And this is the maximum energy that has been used. So that's why you can see here from here to here, so we saved this much of energy. So that's why this is the binding energy, right, for the enzyme and substrate. So we'll use that energy that we saved from the binding energy, right? So that energy is contributed by the formation of the weak non-covalent interaction between the enzyme and the substrate. This is in the transition state, right? And then we go to the final product, that is the enzyme and the product that we get, right? And that's why it uses this much of energy instead of this much of energy in the uncatalyzed reaction, and this is the catalyzed reaction energy that has been used, right? I hope you understand my point. So an enzyme catalyzed reaction is much faster than the uncatalyzed process, right? Because the hill is much smaller, right? So the important principle is that the weak binding interaction between the enzyme and the substrate provide a substantial driving force for enzymatic catalysis, right? So the groups on the substrate that are involved in this weak interaction can be at some distance from the bonds that are broken or changed, and the weak interaction formed only in the transition state are those that make the primary contribution to the catalysis. All right, so now the same binding energy that we saw here that provides energy for catalysis also gives an enzyme a specificity, right? So the same binding energy that provides energy for the catalysis of the reaction also gives an enzyme its specificity. That means the ability to discriminate between a substrate and a competing molecule. If substrate and the other molecule looks the same almost, right? So not it's not like by any chance that the enzyme goes to the other molecule instead of the substrate and it start reacting with them without getting any result. And the specificity why the enzyme is not going to the other molecule and it's just going to the substrate is the same binding energy, right? That helps to discriminate between the substrate and the other molecule. So conceptually, specificity is easy to distinguish from the catalysis, but this distinction is much more difficult to make experimentally because catalysis and specificity arise from the same phenomena. If an enzyme active site, right, there is an enzyme and it has an active site, has a functional group arranged optimally to form a variety of weak interaction with a particular substrate in the transition state, the enzyme will not be able to interact to the same degree with any other molecule. Right, so for example, if the substrate has a hydroxyl group, right, that forms a hydrogen bond with a specific glutamic acid residue on the enzyme, right, any molecule lacking the hydroxyl group at that particular position will be poorer substrate for the enzyme. In addition, any molecule with extra functional group for which enzyme has no pocket or binding site is likely to be excluded from the enzyme. So in general, specificity is derived from the formation of many weak interaction between the enzyme and specific substrate molecules. Right, so the importance of binding energy to catalysis can be readily demonstrated. Right, so in most enzymes, the binding energy used to form the enzyme substrate complex, that is this one here, or this one here you can see, is just one several contributors to overall catalytic mechanism. Once a substrate is bonded to an enzyme, properly positioned catalytic functional group aid in the cleavage and formation of bonds by a variety of mechanism. That includes what type of mechanism? General acid-base catalysis, covalent catalysis, and metal ion catalysis. So these are the distinct from mechanism based on binding energy because they generally involve transient covalent interaction with a substrate or group transferred to or from a substrate, right? So in general acid-base catalysis, so general acid base catalysis. So transfer of a proton is the single most common reaction in the biochemistry, right? One or often many proton transfer occur in the course of most reactions that take place in the cells. Many biochemical reactions occurs through formation of unstable charged intermediate that tend to break down rapidly to their constituent reactant species impeding the forward reaction, right? So the charge intermediates can often be stabilized by the transfer of protons to form a species that breaks down more readily to the products. So these protons are transferred between an enzyme and a substrate or intermediate. Right, so the effect of catalysis by acid and bases are often studied among using a non-enzymatic model reactions in which proton donors or acceptors are either the constituents of water alone or other acidic acids and bases. Right, it's a non-enzymatic model reaction. So the catalysis of the type that uses only the H plus proton or OH, that is the hydroxyl ions present in the water, is referred to as a specific acid-base catalysis. So that uses this, that is this, or OH negative, that's hydroxyl ion. So that is generally referred to as a specific acid-base catalysis, not general, but specific. So this is called specific acid base catalysis, right? So if protons are transferred between the intermediate, that is the substrate, and the water faster than the intermediate breaks down to the reactants, the intermediate is effectively stabilized every time it forms, right? So for example, for example, you can see amide bond, right? Or the hydrolysis of amide bond. So when the amide bond is broken down or its amide is hydrolyzed, right? So what happens? So if if without catalysis, without an unstable intermediate, without the charged intermediate that breaks down rapidly to form reactants. So if protons are transferred between the intermediate and the water, the protons, right? This protons are transferred between the intermediate and the water faster than the intermediate breaks down to the reactants. The intermediate is effectively stabilized every time it forms. No additional catalysis mediated by other proton acceptors or donor will occur. But in many reactions, however, water is not enough to prevent the breakdown to the reactants from the intermediate to reactants. So in these cases, for non-enzymatic reaction, where there is no enzymes in aqueous solution, weak acids and bases can be added to accelerate the reaction rate. So like enzyme accelerate the reaction, but right? But in the non-enzymatic reaction, weak acid and weak base can be helpful to accelerate the reaction rate. So many weak organic acids can supplement water as a proton donors in this situation, or weak organic bases can supplement water as proton acceptors, right? So the term general acid base catalysis refer to the proton transfer mediated by weak acid and bases other than water. That's it. It's nothing. It's not a big deal. So the general acid base catalysis becomes crucial in active site of an enzyme as well. Why? Where water may not be available as a proton donor acceptable. Like there is an enzyme, like there is an enzyme active site. So like there cannot the water cannot reach there, right? So at that active site, the general acid base catalysis become very crucial, right? Several amino acid side chains can and do take on the role of proton donors and acceptors. So this group can precisely positioned in an enzyme active site to allow proton transfer, providing rate enhancement of the order of like 10 raised to 2 to 10 raised to 5. This type of catalysis occurs on a vast majority of enzymes. right? And on your screen, you can see the chart. So the amino acid residues and the general acid forms. That's the proton donor and the proton acceptors. That's the side chain. All right, the second one was covalent catalysis. So in covalent catalysis, a transient covalent bond is formed between the enzyme and the substrate. Consider the hydrolysis of a bond between groups A and B, right? So, for example, A and B, that is hydrolyzed to A plus B, right? That is hydrolyzed, that's it, H2O, right? So, in the presence of covalent catalyst, an enzyme with a nucleophilic group, the reaction becomes something like this. Uh, so, we are doing the A to B, right? And then the enzyme, that is the nucleophilic, that will give you a middle, that is AX plus B, right? And then the hydrolysis of that will give you A plus X plus B, right? So this will be kind of reaction with the enzyme, covalent enzyme or covalent catalyst, that is nucleophilic group. So formation and breakdown of covalent intermediates create a new pathway for the reaction. But catalysis results only when the new pathway has lower the activation energy, right? Than the uncatalyzed pathway, like this. So several amino acid side chains and the functional group of some enzyme cofactors can serve as a nucleophiles in the formation of covalent bond with the substrate. So how this helps? So the one that you saw, all of that also helps in the same covalent catalysis as well. So this covalent complexes always undergo further reaction to regenerate the free enzyme. So the covalent bond that is formed between an enzyme and the substrate can activate a substrate for further reaction in a manner that is usually specific to a particular group or coenzyme. So that is just a covalent catalysis. And the third is the metal ion. Metal ion catalysis. So metals, whether tightly bond to enzyme or taken up from the solution with the substrate, can participate in catalysis in several ways, like ionic interaction between enzyme bond metal and the substrate can help orient the substrate for a reaction, or stabilize charge reaction transition state, or this use of a weak bonding interaction between metal and substrate is similar to the some of the uses of enzyme substrate binding energy, right? So metal can also mediate oxidation reduction reactions as well by reversible changes in the metal ion oxidation state, and nearly all third of all known enzymes require one or more metal ion for the catalytic activity. So most enzyme combine several catalytic strategies to bring about a rate of enzyme, and so these are the different mechanism or like the strategies for the enzyme to activate the catalysis or to increase the rate of reaction. Alright guys, so that was the enzyme biochemistry. So what I was thinking, so we'll continue with this enzyme, but like we'll go slowly with the enzyme because like it has so much things, and like as we are going from the or through the biochemistry, like the chemical formulas, the chemical reactions and all that thing that makes enzymes really a complex topic. So we'll try to do a really slowly and try to grab thoroughly or try to understand thoroughly, and in the next video, so with this enzyme we will continue with the biochemistry, and we'll that is the physiology. The originally we started with the cell, right? So we will be doing cell, but like just before going through the cell and the actual physiology, we just need to understand some basics. So the next topic or in the next video we'll see carbohydrates. Like this is the protein last topic, the enzyme, that's for the protein, and that covers all entire protein. at the end of this enzyme. So we'll just slow down with this enzyme and we'll start with the carbohydrates. So you will see two videos maybe next time, or you will see one video that is with carbohydrates, and next time we'll go with the enzyme, right? So we'll go simultaneously with the enzyme and the carbohydrates, right? So we will be moving further as well, and we're doing the enzymes as well, right? So once we finished carbohydrates, maybe we will be doing DNA and RNA or something like that to understand the basic. And when we finish all that things, we'll jump into again the cell and then the systems. All right. Thank you guys for watching. Right. And don't forget to subscribe to the channel. Thank you.